Welcome to today's webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Shireen Derakshani, and I'll be hosting today's session on starting an antibacterial drug discovery screening program. REVIVE is GARDP's education and outreach program. It aims to connect and support the antimicrobial research and development community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All webinars are recorded and freely available to view after the live broadcast on our website indicated here. As well as webinar recordings, you can also read our series of articles known as Antimicrobial Viewpoints, where experts discuss various topics within their field. We also have a resources section that includes the Antimicrobial Encyclopedia. This has definitions on various important terms, and some of them include videos where experts give a further explanation on the terms. As always, today's presentation is followed by a question and answer session. You can submit your questions at any time during the webinar via the questions window in your webinar control panel as shown on this slide. We will address the questions after the presentation and do our best to respond to as many as possible. Today's webinar is done in collaboration with CC4Carb, the Chemistry Center for Combating Antibiotic Resistant Bacteria. I'm happy to present today's speaker. We have Bruce Blough and Gerry Wright, and our moderator today is Philip Gribbon. Philip is the head of discovery research at the Fraunhofer Institute for Translational Medicine and Pharmacology, ITMP, in Hamburg, Germany. He's involved in several national and European consortia working on compound repurposing applied to infectious and rare diseases. Previously, Philip held various roles in drug discovery at Pfizer, GSK, and more recently at EU OpenScreen, a not-for-profit European research infrastructure consortium. Welcome, Philip. I now hand over to you to introduce our first speaker.
Yep, thank you very much, Shireen, for the kind introduction. So I'm very happy to moderate today's webinar. And our first speaker today is Bruce Blough, and Bruce is a senior research chemist at the CDD at the RTI International, and also he's a principal investigator at CC4Carb. Bruce brings 30 years of experience in drug discovery as a medicinal chemist, and he's led many projects across multiple therapeutic areas, including CNS and also infectious diseases. And that's also included bringing several molecules into clinical trials. Bruce is particularly interested in natural products and microbial signaling, and his experiences covers all phases of the drug discovery and development process, and he certainly can be recognized as a key leader within CDD. Bruce originally graduated from Wake Forest University with degrees in chemistry and followed that up with a PhD at the University of South Carolina. So I'm very happy now to welcome you, Bruce, and the floor is yours.
Oh, thanks, Philip, for the kind introduction. And I'd like to thank everybody at GARDP for providing this opportunity to talk about our program and how we kind of view, or how we're building a gram-negative focused screening library, which is the title of my talk today. Most people don't know RTI. RTI is a not-for-profit institute, a very large one. It has over 6,000 total employees. Most are not in the lab sciences, but the slide you're seeing now is our main campus. It's in between Raleigh, Durham, and Chapel Hill in North Carolina in the RTP area. It was one of the initial seed entities for RTP. My lab is over here on the left side where you can see the building with the hood stacks over here is where I'm at. And our group was one of the original groups here, and we were doing a lot of natural products work, and both Taxol and Camtothecin, two big cancer drugs, were isolated out of our lab here by Monroe Wall. So that's what we're kind of known for here at RTI. And we've since done a lot of steroid work We have a drug in the market, and we have do a lot of CNS pharmacology and chemistry as part of a big NIDA contractor. So that's kind of our background. I'm here to talk about how we're building a gram-negative focused screening library, and it's primarily what we're doing as part of this Chemistry Center for Combating Antibiotic Resistant Bacteria, or CC4CARB. So what is CC4CARB? It is an innovative and unique chemistry center targeting gram-negative antibiotic drug discovery. The goal is to synthesize a library of gram-negative related chemical matter, and to then distribute those compounds to the public for screening or testing. Of course, the purpose is because there's a lack of clinically available gram-negative antibiotics and a pretty lean clinical pipeline moving forward. So there's a need for novel structures in antimicrobials and in gram-negatives especially. And this program is funded by NIH through NIAID. The way RTI views it as it's kind of the front-end discovery piece for the public-private partnership landscape that's emerged over the past 10, 15 years. There's been a lot of work in that area, and kind of it's more development like CARB-X. The biological problem, of course, is with gram-negatives is the cell walls. So there's 2 cell walls for gram-positives, there's 3 for gram-negatives, and the extra layer makes them harder to penetrate and enables a higher level of defense, for example, for efflux pumps inhibitor. So it's gonna efflux pump or in the space. So it's a lot harder to address gram-negative drug discovery. The discovery problems to gram-negatives are kind of twofold. There's been a failure of screening in the system, and there's a lot of natural barriers to novelty. So what I mean by that is big pharma had screened their giant libraries, their 2 million compound libraries for gram-negative antibiotics and failed. And the theory right now is that those libraries didn't contain, they were mammalian focused, so they were targeting mammalian diseases, and weren't really didn't have some of the moieties that you might need to penetrate gram-negative microbes. The one that's popped up early on is if you can go back, is the primary means, and that is through the entry rules that Paul Hergenrother did. And you don't see a lot of primary means in mammalian cell libraries because they're considered a liability. The second barrier is in this field in particular is there's like you need to have understand the mechanism of action pretty early on for something to be taken seriously, and you need to have a lot of safety data and have a therapeutic window pretty early in antimicrobial discovery in order to avoid like something being a bleach or a toxin in the system. So those things you need to have those items before someone takes a scaffold seriously for moving forward. There's also very little funding to move scaffolds forward to sustain fundability. So the solution to this would be CC4CARB, now envisioned by NIAID. Now the idea here is to build a gram-negative focused library by targeting novel scaffolds from individual researchers, kind of the same way that Big Pharma built their libraries. So we are synthesizing small focused libraries for individuals to seed their effort. Hopefully the information we gain can enable their programs to get to sustained fundability, either generating an IP asset for venture capital or generating prelim data for a grant. Then we combine those compounds together to a collection of compounds that could be used as a screening library for other microbes and other related issues involving antimicrobials. And you can schematically see how that would work here. These are different projects all around the screen that could be in this program. You would make different numbers of compounds depending on the project, but when you combine them all, you'd end up with an 800 compound screening library that you can use for gram-negative drug discovery. The overall process that we do is really solicit, synthesize, and distribute for the grant or for the contract We solicit ideas from the community. We then take those ideas and we put them through an approval process, and we synthesize small libraries and then provide those libraries to the contributor who had the idea in the community. The contributor will get about 5 mg per compound. The community will get screening plates in various forms, depending on what they want to do. And that's kind of the whole basic idea about through the CC4CARB process. Scope-wise, we have about 15 FTEs of chemistry, so it's fairly large. It's not huge, but it's pretty big. We are making between 25 and 50 mg per compound in the 25 mg per compound, 25 to 50 mg per compound, and we're fully characterizing them, so it's pretty well done. We are accepting donations and acquisitions into the program now, so we are kind of focusing on getting donations so we can improve diversity of the library. And there is an adjunct testing contract with the library that NIAID added to the program. It's not part of CC4CARB, but it's an adjunct program when funds permit. So we are testing compounds as we get them, as you'll see. The overall proposal process is shown here. Applicant, this is on our website. The applicant will submit into the system. There'll be an administrative review by myself or one of my members of my team. We will have a scientific advisory board review. It's more like NIH review of the program and to see if it fits what we want to do. If accepted by us and the SAB, we then go into a library design phase where we will produce a document called an individual library production plan. It's in collaboration with the applicant or contributor. It's very collaborative in that way. It can be 0% CC4CARB design or 100% CC4CARB design. We've had kind of a mixture of both in the program so far. And these are very flexible. Down the road, if it gets approved in the system, we can adjust it and add or subtract to the compounds that we're making. So that's no big deal there. Once we get the ILPP, it goes to NIAID for review, and then once they approve it, it moves into the queue and we start making compounds and then eventually distribute them. The SIB that we have right now is really good. I really love, I'm thankful, blessed we have this group of researchers. It's Paul Hergenrother, Mike Barbican, Alice Irwin, Andrew Calabrese, and Brian Conlon. They're a fabulous group to work with. They've been really fun to work with. They're very vocal, and they have a lot of comments about each of the proposals, which are all remain confidential, similar to an NIH kind of review. Mike Barbican in particular he's like an encyclopedia of antibiotics, so he knows the history of just about everything that we see, which is really good and exciting. There is an 18-month embargo period involved with this, so we don't disclose anything for 18 months. That embargo period can be extended with approval by NIAID. As far as legality issues, which we get a lot of questions about, the inventorship is out of our hands. It depends on our design involvement per patent law. Ownership is handed to the contributor unless RTI and the contributor have an ongoing or future relationship, and then we kind of have a deal there. But everything gets contributed, turned over to the contributor if we're not involved. So kind of give you an idea of how we've built it, since it's what this is about. Our 5-year, the chemistry portion is 5 years of the contract, then there's 3 years further of distribution of the library. We kind of envision building the system process to do this in about 6 to 12 months. As you can see on the right, at 30 months, where we're at now, it took us about we started synthesis pretty quickly in this program because NIAID gave us some scaffolds that we could start with. The website took about 4 months. We didn't start the SAB until 7 months. And because of the need to go solicit projects, which was the most difficult part of the program to get going and get in place, it took about 13 months or almost a full year before we started reviewing external ideas. And we didn't start synthesizing projects or compounds for that project until about 3 months later. And that's kind of typical once things get reviewed and approved, because you have to go through the review and then go through the design process, which takes a few months to get going. So it took about 4 months longer than we thought, but we are now running pretty wide open, and we have the whole front end portion of it working really, really well. We anticipate about 50 projects. The SAB's reviewed 35, 7 of which were NID. The NID has now approved 27 scaffolds and ILPPs, 3 of which were donations or purchasing ILPPs. But we do have 5 more. 5 more have been accepted and are in the design phase for currently. So we're working on those, and 2 of those should be done actually this week. We anticipate a 6,000 compound library, but we're running behind that because a lot of the projects required a lot of design work and working out the chemistry. And we're also getting some projects that are kind of difficult to synthesize, and I think that's just a matter of companies asking us to do things that weren't working for their internally. We are expecting about half of them to produce some kind of stature. We are seeing probably more than half right now are probably going to produce some kind of stature, and you can see what's already been out there. From a library diversity standpoint, there's a lot of topics in there. You can't really make 6,000 totally unique and diverse compounds because that would be cost prohibited, because they each would have a unique methodology, et cetera. So you have to make sets of compounds. We kind of view the sets as being, you'd rather have 10 analogs of 20 versus over 200 compounds of 1 scaffold, but we have to make these balancing acts because of the way we're constructing it. The main balancing act is between the medchem goals of the contributor and the library goals of NIAID. Contributor goals have, their project goals are less diverse because they're more focused on finding something particular, and a lot of times there's less efficient synthesis involved with that. The library goals are, of course, we want to get more diversity if we can and incorporate high efficiency. So those are always issues that we have to keep an eye on. We do incorporate as much diversity as we can at the beginning before we start going off onto specific lines of inquiry. But we are accepting a lot of scaffolds that aren't well represented in the commercial space, which is really interesting. And we are adding functionalities that aren't well represented in existing libraries in the commercial space, like primary means. And we also hope to include the donations as part of how we incorporate diversity. So that's all ongoing. From a design point of view, we are soliciting unique compounds or scaffolds with gram-negative activity. So that's one way we're getting gram-negative activity into our libraries. We are involved with rescuing known gram-negative scaffolds and some gram-positive scaffolds, for example, overcome resistance. So we're actually incorporating gram-negative compounds that way. We are using the Hergen-Rother entry rules and some of those design principles, like an ionizable nitrogen, low globularity, which is flat, better than a glob of something, and then lower flexibility. So we are incorporating those types of concepts. We are using siderophores. Of course, siderophores are used to hijack the iron sequestration processes like Tone B. We are looking at talking to people that have AI and machine learning capabilities into the program. NIAID specifically asked us to talk to people doing modeling and help them out. It's not very easy to do that because they often want the 200 or 300 unique compounds as well, but we're finding ways to work with them. It's interesting, the way I look at it is it's kind of almost like modeling medicinal chemist intuition, because it's kind of a phenomenological approach to doing it. It's not docking where you're looking at specific structures. So you're kind of taking what the whole community of med chemists might say looks like a bug drug, and you're modeling it into some kind of model, that's the way we kind of look at it, and it's very informative that way. One other thing that's interesting is we see so many projects and ideas coming in that we noticed that there was another moiety, a chemical moiety, that's actually been in, I think, 7 or 8 of our hits that people have given us that are it's also widely avoided in mammalian drug design. And I don't want to talk about what that is quite yet, but we're going to look into it, and it's kind of the advantage of doing this project the way we're doing it is because we have a bird's eye view of a lot of different things, and we can see things like that pop up. So that's a pretty good advantage for doing this. Now I wanted to talk about one project that we are now just starting to talk about, which was vancomycin. So we did do a lot of vancomycin analogs. We had a project doing siderophores, and the data is shown here. As you can see, we looked at quite a few different siderophore groups and linked them through different linkers, and we looked at 4 different positions around vancomycin itself. And you can see that vancomycin is here, and you can see that it was not very active in the bugs we tested for, except for staph. But then when we started looking at siderophores, we picked up Acinetobacter really, really well in this strain. I think this strain might be a little sensitive to activity, but nonetheless, we're picking up Acinetobacter activity, and we're also picked up E. coli activity over here. So we are able to broaden the activity of vancomycin through the use of siderophores. You can see the bugs that we test for. This is a typical bug panel, 6 bugs with 2 efflux pump mutants. And so these compounds will be made available to the public pretty shortly here, once we get the back end piece put in later this year. From a solicitation strategy, that's one of the more difficult parts of the program, and I wanted to address that since it's pretty difficult. We did have a phased marketing approach where we initially targeted proposals, and then we started adding donations to that, and then we're going to market the collection once the library is established and we start making it available to the public. We have slow rolled the marketing once we got up to max, once we got filled up with and running maximum, to avoid a project backup, so we don't have to make too many decisions about priority within the projects. We kind of want to maintain a 20 proposal lead window, so we at least have 20 ideas that we can go out and hit people up with for inclusion in the program. Now, it was really interesting when we started it, who do you actually go and ask for ideas? Because you can't just ask everybody, you have to have some kind of strategy. We actually started with well-established academic researchers. We figured that they would probably have novel ideas that they weren't working on that might be side projects we could help with initially. We also targeted young academic researchers that might need prelim data. So those are the 2 that we mainly looked at from the initial batch. We did do a lot of biotechs marketing and have quite a few of those, which I'll get to in a second. And then we wanted to talk about entities within the community that might have lots of contacts within the community to kind of get the word out. So and kind of spider out from their contacts, which also worked. And so we did get a lot of ideas through all of those. One thing that surprised us was we didn't expect to talk to med chemists very much that had projects ongoing, but it turns out that there's a lot of academic researchers that need help with their projects, and they were viewing us as an extra pair of hands for their projects. So we're talking to some med chemists and have a couple projects where they're using us for that as a resource for their funded academic programs or their projects they're doing from whatever funds they've got. That's an interesting thing we saw. Now, from the tools and strategy, we had to build the website, which was the key tool for this whole project. The website might look similar to another website with CarbX, and that's not a coincidence. We actually developed the CarbX website originally, and we cribbed off of some of those tools to get this started. But we have an external facing portion of the website, which gets into marketing. We can get analytics off of that. It's an interface between the clients and us. But there's also a giant internal facing one that we use to manage the program. So that was pretty key to do everything and get things rolling with the project. We do a lot of general marketing through social media. We had to produce logos, emails, newsletter, brochures, presentations. A lot of pieces were involved with marketing. And we also did a lot of advertising. One thing we found was LinkedIn was a good place to advertise and figure out what advertisements might work well because it's relatively cheap. And then we could figure out which ads work the best there, and then moved it outward to other more expensive places like ACS and AAS. Of course, we do a lot of direct marketing as well with emails and Zoom meetings. We look for academic researchers on publications, grant submissions, and websites, and biotechs on websites. And I already talked about we do go after talk to a lot of the support network people like Mark Blaskovich down in Australia and BARDA. And we have looked for a lot of community members from conference attendance lists and database searches. So there's a lot of legwork involved with all of the solicitation portion of this. It's a very large piece of this program, and RTI had some experience in that previously, which is a good thing. The last couple slides, I just want to talk about some observations we've had with the program. The type of submitters out of the 32 we had 7 biotechs, 15 academics or institutes, including us. RTI has a couple projects in it. And then there were some donations and NIAID projects. The scaffold types, we've seen everything. Small molecules, we've seen known antimicrobial scaffolds that we're trying to broaden activity like vancomycin or get around resistance. We've seen natural products. We've had a couple organometallic projects. Actually, we had one that didn't last very long, but we made a lot of compounds for them. We have a lot of peptides are popping up now, and there are some situations where someone has a technology where they want to alter a compound's properties like penetration efflux. So we are applying that technology to known antibiotics. The stage of development, we've seen a lot of conceptual stuff, which is all the modeling. We want to broaden activity, or you have a virtual hit, but a lot of them are hit to lead or just tweaking SAR. And in general, we're seeing a lot of ideas that we know are difficult to fund elsewhere, and we suspect a lot of things are projects that are difficult to synthesize elsewhere that we're being asked to look into. And the pictures here are some of the scaffolds we've actually looked at as part of the NID set. Finally, in the gram-negative area, what are we looking at? Most of the projects are improving or broadening activity of a known or novel scaffold. We are getting a lot of interest in people developing selective antibiotics, both gram-negative and gram-positive. So, I mean, historically, most people wanted to broaden activity. We're starting to see people want to do selective antimicrobial development, which is probably the future of bug drug development. We have done some efflux pump inhibitor work with a couple people. And as I mentioned, we are applying some novel technology to existing antibiotics. We do get questions about other areas of interest, and we've seen it a lot in TB. Of course, we are biased maybe that way because we're hitting a lot of our early people. We're TB researchers. We are increasingly been asked about non-tuberculous mycobacteria, which is a problem and also has some of the same issues with gram-negatives, including penetration. And we do get a lot of questions about antifungal testing. That seems to be of interest to a lot of people as well. So that's kind of what we're seeing, at least observations from talking to people. We've talked to 50, 60 people at least, I would say, in the field. So there's been a lot of discussions and a lot of presentations given for that. So that's really all I have to talk about. Here's the contact information for us. I'm the principal investigator. Elliot Polly's the solicitation manager. You can see our website link and our email. The NIAID contacts would be Jeremy Starr and Rick Sciatti there at NIAID. They were actually both Pfizer researchers. So that's what I have to talk about. Thank you very much for listening.
Thanks very much, Bruce, for a really informative presentation. Just to remind everybody, we have the question and answer tool, and if you can please add your questions for Bruce's talk now, and then we'll answer them after Gerry's talk. So it's my great pleasure to introduce our second speaker. It's Gerry Wright, who holds the Michael G. DeGroote Chair in Infection and Anti-Infective Research at McMaster University. Gerry's also a Fellow of the Royal Society of Canada, a Fellow of the American Academy of Microbiology, and is a recipient of numerous prizes, including the Murray Award for Career Achievement. He's trained over 75 graduate and postdoc students, 300 manuscripts, so extremely prolific. And his research interests are in the origins and the mechanisms and underlying antibiotic resistance in the clinic and the environment, as well as discovering new anti-infectives. So welcome, Gerry, and we're really looking forward to your talk. So the floor is yours.
Great. Thank you very much, Philip, for that kind introduction, and thank you, GARDP, for putting on this valuable series of Revive lectures. All right, so I'm going to follow up on Bruce's excellent summary of some of the challenges we find here in antibiotic screening. So in order to do an antimicrobial antibiotic screen, what we need is compounds, but also an assay. And so what I thought I would do is sort of go back in time a bit and show you how this area has evolved and where we are now. So shown here is an image of Paul Ehrlich and Sakahiro Hata, who probably did the first screen in looking for anti-infective agents. They were systematically looking for synthetic molecules, dye substances in particular, that could cure an infection of Treponema pallidum, so the organism that causes syphilis, in an animal model. So they did their first screen in an animal, so where the animal was infected and individual compounds were then explored to see whether or not they could overcome the infection. And this is how they found what most people, I think, would agree is the first antibacterial compound that saw clinical efficacy, which is salvarsan, which is this arsenical compound. And as a result, Paul Ehrlich coined this term, the magic bullet. And this occurred about 110, 115 years ago. So there's a long history here. So this is sort of the first foray to looking at systematically exploring the effects of chemicals on microbial activity, but of course using mice or guinea pigs or rabbits in this case as your screening medium is kind of cost prohibitive, and of course you can't go through thousands or millions of compounds this way. Next on the stage was what I would consider maybe the first in vitro screen that is outside of an animal, but of course this was sort of an accidental story. So we know the apocryphal story of Alexander Fleming, who came back from a vacation to find a fungus growing on a petri dish that had strep pneumo on it. Around the fungus there was this zone of inhibition where the cells were dying, in fact lysing. And of course, Fleming actually was also discovered lysozyme, so he recognized this phenotype as a cell lysing phenotype. And that launched the first really dramatically effective antibiotic series, the penicillins, which have, as we all know, quite a storied history and probably responsible for saving hundreds of millions of lives over the last century. So this idea of looking to environmental microbes sort of launched what Kim Lewis has called the Waxman platform, and I think it's a really good way to describe it. So on the left, you'll see Selman Waxman. He was a soil microbiologist at Rutgers University, studying a lot of the actinomycete bacteria that lived in soils. And what he did was sort of systematically exploring these organisms, preparing extracts with his team, and then just systematically testing susceptible bacteria for growth inhibition. And so you can see that you start with a source of these microbes, you isolate the microbes as best as you can, and these days you would array them in some kind of systematic way, like this 96 well plate that I'm showing you here. And you would use these to systematically interrogate the effects of these extracts or synthetic compounds on the cell growth of your favorite microbe, depending on what your TPP, your target product profile is. That's how you would do this. So this approach, and I'm showing you a timeline here that from Lynn Silver's really excellent review about 10 years ago that I encourage you to look at. In fact, a lot of the reviews that Lynn has published over the last decade or so are really informative if you're trying to get into this area. Where you can see that we're, salvarsan here on the left, the discovery by Ehrlich, penicillin, and then when the Waxman platform sort of gets systematically applied in drug companies around the world, we have what is termed the golden age of antibiotic discovery. The majority of chemical scaffolds that we have in use today in the antibiotic field were discovered then using this very simple systematic screen. And after that, there was a lot fewer discoveries of new kinds of molecules. And in fact, that although we still have a lot of tremendous activity in antibiotic discovery, but mostly through medicinal chemistry on these known scaffolds like penicillins and cephalosporins and rifamycins, for example. And after that, there's been this, as we all are aware of, in fact, one of the reasons why GARP exists is there's been this really significant challenge in discovering new molecules, and this is what Lynn has called the discovery void. So how do you do this screen in practice? So these days, in Waxman's day, you would sprinkle these organisms on a petri dish and see what happens. But these days, you want to do things in much higher throughput, and so the advent of automated robotic systems and multi-well microtiter plates like these enables you to be able to interrogate a number of different either synthetic compounds or natural products or semi-synthetic versions of those, depending on what your interests are, in a very high throughput fashion. So you can use 96 well plates, 384 well plates, even 1536 well plates. And this goes up even further to like 62, 52 well plates, or not well plates, but rather arrays on solid media where you can actually pin even 4 times this density on the same area of agar. So the strategy is actually incredibly simple. You add compounds, you add media, liquid media, for example, it might be your favorite one. Mueller-Hinton broth is used a lot because that's what's the standard for clinical MIC determinations. Add your organism of choice, incubate for an appropriate amount of time at an appropriate temperature, and then you just read out your plates. For a very simple growth inhibition assay, you could just use the optical density at 600 nanometers, which just looks at the interference of growing cells with white light at that stage. You can use fluorescently tagged microorganisms that allows you to look at things much sooner and then even in real time. Luminescence is exactly the same strategy as well. So there's lots of different ways that you can modify this system depending on the microorganisms that you're looking at and maybe how long they're growing. If you're doing this, for example, with Mycobacterium tuberculosis that takes days to grow, you might not want to use the optical density at 600, but you might want to use a luminescence or fluorescence readout. There's variations on this theme. I indicated liquid media, but you could definitely use solid media, which impregnated at this time with the bacteria, and then you pin compounds on top of it. So lots of different strategies can be used to this, but the approach is very similar to that which was developed by Waxman, in the 1930s and 40s. So we call these whole cell screens, and they work incredibly well. And in fact, I dare say most of perhaps all of the antibiotics that we have in the clinic right now were discovered using this strategy. And it continues to work to this day. So here's just 2 examples straight out of the literature this summer. In this one that was published in Cells online, as you can see it from a collaboration again with Kim Lewis, the identification of an antibiotic from a so-called unculturable bacterium, but really an organism that is rare in the environment. So one of the challenges of screening natural product producing organisms like Waxman did is that it's easy to find the same molecules over and over again. So if you want to find rare molecules, you need to look oftentimes to rare microorganisms. And here's an example where that worked out well. So again, the screen that identified this molecule was very similar to what I just described, and it has this very interesting mode of action where it interacts with lipid 2, lipid 1, and undecaprenyl pyrophosphate. So 3 different targets that are hard to mutate, and that makes that a sort of resistance light kind of antibiotic. Another example of applying sort of modern technology to this screening approach is one from my colleague down the hall, John Stokes, where you're using artificial intelligence, machine learning, training sets. Here in particular, they're looking for compounds that are specifically targeted towards Acinetobacter baumannii. So you train the set on screens of Acinetobacter, E. coli, and Staph aureus, the computer figures out what differentiates them, and then you predict structures and then either synthesize them or access them in a way, and again, screen them in exactly the same way that I just mentioned to you. So that's whole cell screens. It kind of limits you though, and we've realized this over time that using this approach, we often use pretty rich media. And that is because we're all impatient and we want the bacteria to grow fast. And we want to give them all the necessary nutrients to do that. But of course, this misses what it's actually like for an organism to be in humans or an animal or whatever, where it's actually causing an infection. So there, the conditions are much more stringent for bacterial cell growth or fungal cell growth. This strategy approach is equally applicable to antifungals. So it misses things like antimetabolites or inhibitors of virulence. And here's, and a great example of this is that the second sort of antibacterial compound that was broadly available to people were the sulfa drugs that were originally discovered by folks like Erhard Tomak in the German chemical industry, where again, compounds like this, protonsil, were discovered by screening in mice. And the reason why you could figure these things out that this was available is because this is an antimetabolite, and so you only really get the efficacy in conditions of infection. So ways around that now are to screen in media that better mimic the case during infection. The downside of this, of course, is that it takes longer oftentimes for the bacteria to grow. But if you screen in minimal media, for example, you can catch antimetabolites, or if you screen under more physiologic conditions, you can start to narrow the target space to those targets that are really only essential during infection. So, for example, again, my colleague Eric Brown down the hall from me did, I thought, a really nice job of systematically approaching this in Klebsiella pneumonia. So, in high nutrient broth, wild type organisms or ones where we've had a series of mutations being introduced in a variety of open reading frames grow equally well. But in serum, what you see is the wild type grows nicely, but the mutant does not. So this tells you that this gene where this mutation has occurred is essential for its life during an infection, while it's living in serum. So you can recapitulate this idea using compounds. So screen in nutrient broth or in serum, and these compounds like this, where you see a nice dose dependence in serum, but nothing in nutrient broth, tell you that these compounds are targeted towards targets, molecular targets that are important during infection. And these can be things like antimetabolites, as we discussed before, similar to the sulfa drugs. Or importantly, they can be things where the host actually participates in activating the molecule of interest. So things like prodrugs that are activated in serum. And you wouldn't be able to see this in a normal sort of screen that I just showed you before. So there's tremendous advantages to this kind of strategy. And then you can also find unusual molecules like these organometallics that really only show activity in vitro settings. So lots of different ways to approach this to ensure that you increase your hit rate, moving beyond the sort of traditional Waxman platform. So if we go back to this slide that I showed you, we have this discovery void, and part of the reason for this is that a lot of the antibiotics that were discovered are products of soil microorganisms or fungi. We kind of picked all the low-hanging fruit and ran out of new chemical matter around the mid-1980s. But interestingly, at this very same time that platform started to not be as productive as it used to be, we saw several other technological advances. We saw the first sequencing of entire genomes of microorganisms. We saw the advent of combinatorial chemistry to increase the number of compounds you have. And importantly, we saw huge changes in the way that you could approach the biochemistry of specific targets with protein overexpression, ready access to three-dimensional structures of proteins, for example. It's worth noting that prior to roughly the mid-1990s, if you got a crystal structure of any protein, you were probably on your way to get a paper in Cell Science or Nature, because it was so hard and so rare to get. But once we started developing high throughput methods to be able to do that, and new technologies such as the use of synchrotron radiation, those became much more common. So you have all these things happening at the same time. And so there was a switch in mindset from in the pharmaceutical industry, in drug discovery as a whole, not just in antibacterial discovery, that maybe the way forward is to take full advantage of this and not use wholesale screening like we did in the past, but use target-based screening. So here the idea is that you have your favorite target that you've identified as being important during, to cell growth, for example, or during infection. You would screen, as Bruce noted, millions of compounds in your library that you've mastered, that you've synthesized, or that you've purchased, or that you've collaborated with. We've got robots now that can do all of these things, and then look for molecules that engage the target in this fashion. So this just recaps that. These target-based screening approaches, TBSs, and lots of effort was made in the 1990s and early 2000s to identify that critical set of essential proteins or receptors in both human biology, but also in bacteria, and to screen these using the available chemical libraries in-house, thinking that this was going to be their way forward to find new antibiotics. So this was, like all things in the world, there's change happens because of changes in fashion, and most of the pharma industry moved to this approach. And what were the results? Well, GSK reported in this really nice paper, and I encourage you to, if you're interested in this field, to be looking at it, to look at this paper. They did 70 screens between 1995 and 2001 and found absolutely no candidates. this same, the same exact thing happened with Astra. So two large pharmaceutical companies, target-based screening approaches using purified in vitro systems with the exact same result. And there's a lot of great lessons to be read out in these papers. So why is that? And Bruce alluded to that. The challenge is that bacteria have been dealing with molecules, small molecules, for billions of years. They're well armed and well protected against them. And in so gram-negative bacteria, for example, you have an outer membrane that has physical chemical properties, an inner phospholipid leaflet, and an outer lipopolysaccharide leaflet. You've got that has its own physical chemical properties. You then have to go through an inner membrane if you're trying to access, for example, an essential enzyme inside the cell, a phospholipid bilayer with its own different chemical properties. This outer membrane is highly impenetrable to many different molecules, and so you have to get things through these protein gates called porins, and then when the next thing that they encounter are these vacuum systems, these efflux pumps that bacteria have evolved and have redundant backup systems and a lot of diversity for them to be able to basically rid themselves of noxious compounds. So very, very challenging. If you compare this to a human cell, for example, I mean, human cells are wimpy compared to these. These are well-seasoned, highly evolved structures that are a major challenge in antibiotic discovery. So, and as Bruce noted as well, we had not only this, we have this challenge. Most of the chemical libraries that were screened in antibacterial campaigns were based on physical chemical properties that give you good drugs for humans, and not necessarily antibiotics. And so, for example, those that adhere to Lipinski's rules. If you're familiar with those, like Lipinski Rule 5, and very importantly, you need to remember that Lipinski's original paper said these rules work pretty well, except for antibiotics. So if your chemical libraries, your millions of compounds adhere to those rule sets, you're fishing in the wrong pond. And at the end of the day, the other antibiotics were so amazingly successful. They were so safe, so non-toxic. Even if you found a compound that could get through these barriers and could do everything, the toxicity threshold that you wouldn't worry about if you were making a cancer drug, for example, was really something important to consider in this field. So making antibiotics is very hard. Making drugs in general is hard, but making antibiotics is doubly hard. All right, so just a little recap here. So the pros and cons of target-based screening versus whole cell screening. Target-based allows you to focus on your favorite target right up front. That means that you can move very quickly in cycles of SAR and target engagement to get better inhibitors or tighter binders or whatever it is using modern biochemical techniques. The challenge is, of course, we have no idea if it's actually going to work in a cell, and so you need to build that into the program right away. And at the end of the day, it has a terrible track record in this field. Doesn't mean it's not going to work. And I think it's really important to realize that these strategies should always be on the table, but it's challenging to make work. Whole cell screening, on the other hand, excuse me, has a great track record. Things that are active in this screen by definition are bioactive, but you really need to work hard after that to figure out what the target is. And many times the target is really difficult to find. And if that's the case, then oftentimes these molecules are quite toxic. So that might not be easily used to develop or improve your hit to lead status using a systematic fashion. There's a way that you can combine sort of the best of two worlds, and that is you can use target-based whole cell screening. The way you would do that is you would find your favorite target. And here's, I'm going to show you an example from the Merck team, where they're really interested in the enzymes involved in fatty acid biosynthesis. And what you can do is using antisense technology is decrease the expression of your target genes. And if your antibiotic or your molecule of interest will engage that target in that screen, you become hypersensitive to the target, right? So here's an example where they've done pinning of molecules on an agar plate. And you can see these zones of inhibition, right? These things are toxic to the bacteria. This is the control, so lots of hits here. And what you can see is if you knock down this specific RNA that encodes FAB, which is essential in fatty acid biosynthesis, you get an extra one that doesn't appear here. So B6, you can see now has a zone of inhibition. And that turns out to be, in this proof of concept story, serolonin, which is a known inhibitor of this system. So you can combine, the sort of the best of both worlds. Another target-based whole cell screen is the target resistance itself. And this has been done incredibly successfully using inhibitors of beta-lactamases. And so first, the first natural product to do so, clavulanic acid, discovered in the early 80s, and then followed up by some synthetic derivatives. Most of the new antibiotics that have come to market over the last little while are combinations of beta-lactamase inhibitors and beta-lactam antibiotics. You could think about this in any kind of resistance inhibition, so efflux as well. So far there hasn't been any successful examples of that, but there's no reason to think that that's impossible to do. They're just challenging. So to address this, my lab has built a platform to help with this, where we can have an isogenic series of E. coli, either wild type or permeable E. coli, where we've individually added single resistance elements suitable for screening. The platform has about 120 different flavors of resistance, and you can screen for whatever it is you want. And here's an example where we did a screen of this E. coli system expressing NDM1, the metallobeta-lactamase that inhibits carbapenems. And so what we're looking for is regain of activity, and using a natural product extract screen, we identified this molecule that selectively binds zinc, and it works really well in animal models of infection, and actually went down the preclinical development pipeline quite extensively over the next few years. And then just finally, you can go back to the idea of screening inside an animal. So screening inside of mice or rabbits or guinea pigs is cost prohibitive and probably unethical. But you can do this in high throughput with other animal models. And here's a great example pioneered by folks at Harvard, where using the worms, C. elegans, as that surrogate animal. And so what we're looking, in this case, we grow the worms, synchronize them to a selective growth stage, add the media and compounds to the plates, as we discussed before, infect these worms with Enterococcus faecalis, a common human pathogen, transfer the worms to microtiter plates, let them grow, and then image them. And what happens is that if you've got an antibiotic, if you don't have an antibiotic, the Enterococcus kills the C. elegans, and they appear as these little sticks here, very unhealthy looking C. elegans. But if you have an antibiotic, in this case ampicillin, that kills the Enterococcus, and the worms have their natural shape, which is all these little curvy, lovely little diagrams. And you can do this in high throughput and look at thousands of molecules that help you screen in an animal to identify compounds that perhaps are de-risked a bit more down the pipeline. So both whole cell and target-based screens have their advantages and disadvantages. As I noted, these in vitro, I'm sorry, that's a spelling mistake here, in vitro, so purified protein target-based screens have not yet identified any new antibiotics that have made it to the clinic, but they might. And I think if you combine these strategies together, perhaps in an animal model, you can really start to take advantage of innovation that's occurring in a number of different fields. So just a final acknowledgement of stuff that goes on in my lab is funded primarily by Canadian funding institutions. This is my team, all of them working on different kinds of antibiotics and antibiotic discovery. And I'll finish off there. And again, thank the organizers for asking me to give this mini lecture.
Yeah, thanks very much, Gerry. That was a really exciting talk. I think highly complementary to Bruce's talk about the various libraries that are open to us. So yeah, it was really nice. So as you can see for the audience, as you can see on your screen now, there's an opportunity to ask some questions, and we'd like you to use the question bar there. We've got several questions in there already, but please use that to ask some questions now, if you can. So there's some questions come through already for Bruce, and maybe I will sort of start off with this one. And I also had a question around it, but one of the attendees is asking what the capabilities are for synthesis of natural products within the platform. And I guess I have an add-on for that is, what about the submission of natural products as well? So how do you deal with natural products in general?
So we are, RTI was a natural product lab, so we have a lot of experience historically with natural products. We can handle some things. We actually have a project where we're synthesizing a de novo, a natural product. So it's not, we can handle it if it's not too extensive. We can't do a 20-step synthesis. That's a little bit too much for us probably, but this particular one is 5 steps. So we can handle something like that. We are also, though, one of the projects we have is a pretty complicated macrolide that comes out of a fermentation broth. And originally the submitter was doing the fermentation themselves and sending the compound to us, and then we were making analogs off the natural product like we would normally do here. I mean, that's the first thing we would do here if we got a natural product, is just start what chemistry works, what doesn't, how to make analogs, that kind of thing. His speed was kind of a little bit slower than we wanted, so we're actually have employed a firm to do fermentation of that broth within the program now. So we're scaling that up ourselves, so we can do that kind of thing. So we can modify existing natural products if it's more difficult or complicated. If it is something simple, though, we can reach it, then we can do one-off syntheses of that for sure.
Okay, thanks, Bruce. So there's a question for Gerry around biofilms and the observation that with AMR's pathogenesis is associated with biofilm formation. So how can you screen in the presence of biofilms? What are the technical solutions there?
Yeah, so you can do that. You can screen specifically for either inhibitors of biofilm formation using what's called a Calgary pin test, I think, or something like that. So basically, it's a device that you put on top of your 96 well plates that actually is like a little plastic pin that goes into the solution of where the bacteria are growing, and the biofilm gets formed on the outside of that. You can visualize that and quantify it just by staining it. So you can look for things that actually inhibit biofilms, or you can look at things that dissolve biofilms that once they've already been made, and there's ways that you can do that too. So these strategies are very amenable to that. A lot of antibiotics actually at lower doses induce the production of biofilms because the bacteria get stressed and they run for the I think the closest surface they can find, and you can actually use that to find low concentrations of antibiotics too. My colleague Laurie Burrows has done that. So you can adopt these strategies for biofilms, either biofilm formation inhibitors or biofilm dissolution molecules. The question is always, they're really important, but is there a market for something that just does biofilms? And I'm not sure. There might be, but it's just something to consider all the time when you're doing these. Is there a way to move your product forward if you're considering commercialization?
Thanks for that. Bruce, there's a slight variant on the natural products question. Is that one of the attendees is asking about an efflux pump inhibitor, which is being derived from a plant natural product, and asking are they sort of classes of natural product which you see as promising leads for efflux pump inhibition based upon the work so far?
Nothing so far that jumps out as a class, I would say, but we just have a couple projects in that, for starting out, so we don't know, and we're actually not testing within the program for efflux pump. So anything that would come in, the contributor have to test for the efflux pump inhibition. So we're not doing any kind of combination testing quite yet, unless we have extra slots available for that. But we haven't seen anything quite yet.
Okay.
We do have one idea it's a very small fragment that looks like it's really good at blocking efflux, and we're thinking about talking to someone about actually linking that on to known antibiotics to see if we can block it with just that little fragment hanging off of another known antibiotic. So it's not something that's giant like polymyxin or like that, but it's a very tiny group.
Yeah.
That's pretty intriguing to me.
Okay, so thanks for that. Also, Bruce, it looks like we have a potential customer for the platform, and somebody has some new natural products, and obviously they consider that they're going to need to bring along some validating bioassay data prior to be considered, these to be considered as eligible scaffolds. So what's the minimum data set, or the minimum bioassay data set that you need? I know you mentioned you were getting outcomes of AI and other sort of computer generated results, but do you need to bring in biological data as well?
In some cases where it makes sense that we need that, yes. So if you're pitching something that's you have some resistance kind of thing, and you're actually you need to show it, but we're not requiring it. We'd like to see it as much as we can, but it's not required, because ultimately we're trying to make a library of gram-negative related chemical matter, and if it's a novel structure that isn't well represented in the commercial libraries, then that represents some space that we could get into. And we would likely make analogs with, siderophores in it. You put some primary amines, guanidines, those kind of things on the structure if we don't know it very well. So we are amenable to do that. The information in the proposal process is we made it as minimal as we could because we want to facilitate ideas coming in. We knew there were people that were not chemists would submit ideas, so we want to make sure we can keep it as easy as we could. We do encourage everybody to talk to us ahead of time, because you can submit the proposal on the website electronically. It's a one-pass proposal submission, though we did that so we didn't have to create username and passwords that would create a security issue with our system. So there's also instead there's a Word document you can download off the website and fill out there and send it to us, send the idea to us on that, which keeps it confidential within the system. And then we can review it ahead of time and just discuss it. We do that often now. We kind of help people go through it and say, is this enough data? Is it not enough data? We can answer all those questions prior to submission, basically through the process. We'd be happy to do that. So whoever it is, just contact us.
Sounds good. So, Gerry, I have a question asking for your thoughts on the usefulness of isolation chips in discovering novel antibiotics. Is that?
You mean for the enrichment of rare microorganisms?
I think that's where they're coming from. Yeah.
Yeah, I mean, it works. There are rare organisms out there, and and I think the clovibactin story that just came out in Cell is a good example of something that is not commonly found. The challenge when you're just taking regular environmental samples is that the weeds grow first, and the weeds have already been screened, and so getting rid of the weeds is important. Finding different ways to do that, like the ITMP or pre-treatment with high doses of antibiotics or other kinds of ideas, allow you to enrich for microbes that are otherwise easily missed because they get outcompeted so easily in the lab. So ITMP is an interesting technology. We haven't had any success in trying to make it work well, but others have, and it's probably just a technical glitch. So I think anyone who says that there's only one way to do this is incorrect. Nobody knows.
Yeah.
You should try everything.
Yeah. So it's a question for Bruce around the sort of long-term availability of a library and what the plans are. So once you get to the 6,000 compounds, is it envisaged that you would sort of continue to sort of hand that out on a case-by-case basis, or will that be available as a sort of a commercial set via one of the sort of classical providers? Do you have a model for that yet?
Yeah, so we're going to start rolling the library as we have it out later this year. We were trying to develop the infrastructure for pushing it out and trying to decide how to do that, which is exactly what we're talking about. It's going to come in two ways. Because we're testing compounds and we've got some hits and active compounds, we're going to provide us a cherry-picked set of compounds out of the libraries that pretty much were active compounds somewhere else in one of our screens, just to, because under the theory that if it's active somewhere already, it might be more active down the road, and we don't know if some groups don't have the resources to screen thousands and thousands of compounds very quickly. So we can provide that. That's one format. The other format will be each library that we produce for each of the projects will be rolled out as we release them and release the data. So everything will be published on our website. We're working on a PubChem interface right now that's kind of searchable, so to be structure searchable on our website. And you'll be able to request individual compounds if you need them, but we would rather prefer them being provided in plates for people, enough to do initial screens. And then if they have hits or they want to follow up on, contact us again, and we can provide a larger amount of material. Because it's amazing how fast 20 milligrams doesn't last as long as you think it does in the library production world. We can tell you that. Yeah. So.
Okay, yeah, thanks. And a question for Bruce around the sort of AI, sorry, not for Gerry, around the AI models. And are the sort of data from whole cell screens able to sort of train AI models when you see these contributions of effects like prodrugs and antimetabolites? Are the models able to actually predict those, or are the training data sets sufficiently broad and well annotated so that those types of drugs will come out of these machine-based models, or are they always just going to be arising from serendipity?
Well, I'm not. So the best available data sets are for AI right now are those that are involved in just sort of in vitro whole cell screens in media, as opposed to in serum or things like that. But there's no reason at all to think that you couldn't do that with a large enough training set under a specific condition. The examples that I'm aware of in using AI right now have used training sets that use either generated data within the lab or data that's available publicly through some of the publicly large-scale screening campaigns. And of course, the more molecules you have in the training set, the better chances are you have of predicting. And so it's just ramping up those data sets for things like serum screening and stuff like that to be able to inform the models better. But there's no reason why they can't.
Yeah. Okay, thanks. And Bruce, there's a question on your sort of design rules and the entry rules from the Hergenrother group is one of the elements in your gram-negative design. And the question is, have you thought of incorporating other property rules for bacterial species beyond E. coli, for which the original entry rules were originally worked out for?
We've thought about doing that. We have looked into some of those other, physical chemical property kind of rules, but I don't know we have so many diverse structures. I don't know if generalizing is as productive as people might think. I mean, I know Gerry just said if you think one way of doing something isn't really correct, you could say the same thing about a lot of these other issues. I mean, sometimes those Hergenrother rules work and sometimes they don't as much as you would think. So, but we do have thought about generating some based on basically when we start getting some of these better hits and larger sets of compounds, start generating our own models for sure. And I will say one funny reactions with the AIs are our SAB when they saw the first AI project come in, they would go, well, making those compounds is obvious. That's what we would do, right? Why do we need AI? So there was a little bit of a reaction to AI because a lot of it's phenomenological, what you're looking at, right? And that's why I realized it looks more like what a med chemist intuition would do for some of these projects, but it's more detailed. I will say that.
Yeah. And we have another question around the sort of design of the library, and really have you done I guess you're only 2,000 compounds in or so now, but have you done any comparisons with sort of pre-existing libraries? And you know, as Gerry showed, the sort of very low success rate within the pharma companies of those libraries. So have you done a side-by-side comparison with other synthetic libraries and other antibacterial libraries that are offered by various commercial organizations?
Yeah, we've looked at those, and we try to stay, as I noted, we try to stay, include compounds that aren't in those libraries. So we try to maintain a distance from the commercial space as much as we can. Some of the projects we're doing, you can't do that because you're working on known antibiotics, and some of the big, actually several of the ones that Gerry mentioned we're actually working on with different people, but they're going to end up overlapping it somewhat. But we're trying to create as much diversity from the commercial space as we can.
Yeah, thanks. It's another question around the sort of hit rates that we see with sort of target-directed wholesale screens, Gerry. I mean, typically they're an order of magnitude lower than you get in a wholesale screen. And does that give you any? Is it good in the sense that there's probably less artifactual compounds in there, or is it bad in the sense that you don't have many starting points? Or what does it tell you about druggability? So any comments on hit rates in those types of screens?
Yeah, so if you do a cell-based target screen like the one that I showed you, you by definition are narrowing the target space, and the hit rate will decrease. So you have to have a compelling reason to do that. There's oftentimes good ones. If you saw that image that I showed in the slide from the Merck team, there are a lot of hits that weren't on that specific target. And the question is, are they good? Are they good hits that you're losing, or are they just, as I think Bruce used the term, bleach? And so there's, in particular, when you're screening, natural products, there's a lot of toxic molecules that have been evolved over the last several billion years, and bacteria and the fungi who make them don't really care, how they work, but you might because they're making them for their own reasons as opposed to making them as drugs. So the hit rate by definition will go down, but if you have a compelling reason, if your company is really invested in a certain class of target and you know that you can drug that target, finding those molecules this way is an excellent idea.
Yep. I think we have another probably quite big question, but how would you, to Gerry, how would you design the perfect in vitro wholesale screen? And do you look for compounds that lead to, let's say, the downregulation of your target by some kind of modulation of transcriptional effects, or are you looking for compounds that directly inhibit an enzyme or some other functional protein? So, I mean, is that, and it's a big question probably, but
yeah. Well, again, I think it really depends on what the expertise of the team is, right? Like if you go in, being target agnostic, then you need to be able to have the skills to be able to address all of those targets in some way, or have a connection or a team of people that can help you move that forward. Otherwise, it's probably wiser to go after something that's particularly where you have some particular expertise at, unless, again, you know, you're Roche, where you've got armies of people and you can collaborate with people around the world who are experts once you figure out what your target is, as long as it doesn't, you know, kill people. So it's a huge philosophical question. It really is embedded in the team and the goals of the team. You know, one of the things I've learned, you know, being in this field for this time, and I bet you Bruce would agree, is that, you know, teams often fail because they haven't really understood where their skills are and their target product profile might be. What exactly are they trying to do? It's easy to find molecules that kill bacteria. It's really, really hard to turn those molecules into something clinically useful. So you really have to understand who will use it, who's your end user, what are you trying to solve, all that kind of thing together, and then sort of work back from that and figure out what you're good at. At least that's my thought.
Yeah. So I just the final question for Bruce. So is the platform open only to US-based organizations, or is it something that we here in Europe or elsewhere could access those resources? Is there a policy there?
No, it's international. We have quite a few people around the world. We've worked with people in Sweden, England, Spain, China, India. So we work globally, and the compounds will be available globally as well. Although we're a little worried about shipping plates around the world. I'm not sure how that's going to work quite yet, because they're going to thaw out if they get stuck in customs too long. But we'll have to deal with that when we get to it.
Yeah, that's been one of the side effects of Brexit. We've also seen here in Europe. But yeah, that sounds good. So I think I'd like to sort of I think we've come to the end of the Q&A part, and thanks for the sort of excellent questions. I want to thank everybody for giving the questions and also their sort of comments on that, and especially both our speakers for two really excellent and highly informative talks that will get me thinking certainly to what we do in our next antimicrobial and antibiotic programs here. So back to you, Shireen.
Thank you, Philip, for moderating today's webinar. And of course, thank you very much, Bruce and Gerry, for your presentations. I'm very excited to invite you also to register for our upcoming webinars later this month. You can find the registration links and further detail on the Revive webinar page of our website. And also, please make sure to keep an eye out for our newsletter, and also follow GuardP on Twitter and LinkedIn to stay up to date. So that's all for me. Thank you everyone for joining today, for contributing to the discussions. I hope you found the webinar interesting and useful, and that you will join us again in the future. Please do spread the words. Encourage your colleagues to join as well. Thank you, everyone. Goodbye.